Dr. Bowman, welcome to the show. It's great to have you here.
Thanks, it's a pleasure.
So, Dr. Bowman, tell us a little bit about yourself and a little bit about the project you're working on.
Yeah, I've had a really long-standing interest in biomedical research, having lost most of my vision to a juvenile form of macular degeneration as a child. And about the time that the implications of that started to dawn on me was right as the human genome was being sequenced and Dolly the sheep was being cloned. And so I got really excited about the potential to be a part of this effort to understand complex biological systems like ourselves and how we can control and fix them. I've been involved in this track for a long time, kind of did a create your own computational biophysics degree as an undergraduate student at Cornell, and then went off to graduate work at Stanford, and now I'm on the stint at Berkeley as a fellow, and then I'm on the faculty at the Washington University School of Medicine now, where I'm continuing to work on understanding protein dynamics and implications for drug design, for example.
So you're working on particularly, folding at home. And so if you were to sort of explain folding at home to, your grandmother or a child, like how would you frame it so that everybody understands what folding at home is and what it does?
Mm-hmm. Yeah, great question. So folding at home is basically a big distributed computing cluster with the primary focus of understanding all the moving components of a protein and how they contribute to its function and malfunction in diseases like Alzheimer's disease and cancer, and how we can use this information to design new therapeutics. And ultimately, one of our major objectives is to use the now millions of computers around the world that are volunteering to run simulations for us, using all of this to build a map of the different structures that a protein adopts and how the protein can hop between them. And the map analogy is a good one. We can't just take a satellite image of this space that we're trying to explore. So essentially what we're doing with all of our volunteers is sending them out like cars that are driving around their local vicinity and sending us back the GPS coordinates. And then on our side, we piece this all together into a map that extends far beyond the reach of what any individual explorer did.
So basically, you're just using the power of everybody else's computer to supercharge the work that you're doing.
That's right, exactly.
So how would you put that into some sort of measurement that someone could get their head around? In other words, is that equal to an IBM supercomputer? Is that equal to, how would you compare that to, how we would think about spinning up systems on Amazon?
Mm-hmm, mm-hmm.
How would you make a comparison?
There's a couple different perspectives one can take. So one is the peak performance in terms of number of computations we perform per second. So our estimate right now is that with all the volunteers that we've pooled together, we are the first computer to reach the exascale. So that's a unit of measure. Currently, the world's fastest supercomputers are measured in petaflops instead of exaflops. So our estimate is that our peak performance is about 10-fold that of the world's fastest traditional supercomputer. So this is an enormous computational resource. And even before the 100-fold growth that we've experienced since starting our work on COVID-19, we were routinely running calculations that would have cost millions of dollars to run on the cloud. So now 100-fold increase in compute power, we're talking about hundreds of millions of dollars or more worth of simulations.
Now that's so you've got to use these systems when people make them available. So does that change the time it takes to do this?
Mm-hmm. Yeah, so it's a really interesting paradigm, right? Because normally on a traditional supercomputer, one is thinking about how do you get all these tightly integrated computers working together on what is essentially one big calculation.
Right.
And our whole approach is how do you take this one big calculation and break it up into pieces that can be performed completely independently of one another with no communication between these computers. And so this has been really fascinating because it allows a tremendous level of scaling, and we get a bunch of features like fault tolerance for free. So if someone turns off their machine or their hard drive dies or whatever, it slows us down a tiny bit, but we just keep crunching along. Whereas failures like that can be very hard to deal with in a traditional supercomputing setting.
Right, right, right. So before the COVID-19 pandemic, so what were you guys working on?
Yeah, so we've got a number of research labs that are involved in folding at home, and each of us is tackling a number of problems. So prior to the pandemic, my group alone was doing a lot with Alzheimer's disease and Ebola virus and antibiotic-resistant infections, each of which can be thought as a global pandemic in its own right. We've done a lot with cancer collectively as well, multiple forms of cancer. So there's quite a broad range of biomedical problems, as well as very basic questions like the original project that Folding at Home focused on, which was how do proteins fold into these functional structures. So that the breadth is really a fun part of this, and the generality of this platform. But for the time being, we've put basically everything else on hold, and we're bringing everything we've got to bear on COVID-19.
So, explain, using COVID-19 as an example, sort of explain the science behind, folding at home and what you're specifically trying to do with COVID-19.
Mm-hmm. So one of the challenges at the length scales involved with proteins is that we can't just zoom in on them with a really powerful microscope and watch what's happening on these scales. Everything is very indirect. And so one of the consequences of this from an experimental perspective is that we can usually, at best, build a model of what a protein usually looks like, right? And this is immensely valuable information, but it's also far from complete. So taking American football as an example, if you ask what do the players usually look like, it's them lined up at the line of scrimmage, waiting
Right.
for the next play to start. And there's a lot of information there, but really what you want to see is the game unfold, right? To see who's actually going to win and what strategy should you employ. So what we're doing with our computer simulations is filling in all of these moving parts and seeing what we can learn from that about how a protein functions or malfunctions, and how one could get in there with drug design, for example, to fix problems. So with COVID-19 as a specific example, when you see these pictures of the virus with all of these often red protrusions sticking off of the surface, each of those is a complex of 3 proteins called the spike. And you always see them in those images in this closed conformation where they're packed up against each other. And one of the really cool things here is that one of the ways the virus evades an immune response is that in this closed state, the interface of that set of proteins that actually binds to a human cell and initiates infection is closed up and protected from being recognized by the immune system. And so for the virus to infect us, this thing has to open up like the mouth of some
Right.
Demogorgon monster and in order to expose that surface and make it accessible to engage with a human cell. And little is known about the open state or how this process of opening occurs. And we're now able to capture that with Folding at Home and start exploring whether the different stages of this motion might serve as valuable drug targets, for example. And going beyond the spike, now we're looking at basically every protein from the virus that we can build a reasonable starting model for, and hunting for moving parts like this that might be interesting drug targets.
So if you were to describe some, maybe in more detail, like what a proposed solution is that you might be able to bring to the table with the technology that you've got, like how would you describe that to someone?
We've got a bunch of things going in parallel right now. This is one of the really exciting things, is that we've very much switched from a mindset of what should we do to make the best use of this limited resource to what else should we be doing?
Right.
There's so much breadth of compute power. So we've got a number of things going. One of the things that has the greatest potential for impact on a short time scale is that we're performing enormous computational screens of large libraries of chemical compounds searching for those that are likely to bind most tightly to some of the key components of the virus, like the main protease, which is one of the popular targets for drug discovery. And through a project called the COVID Moonshot, we're teamed up with a number of experimental groups who are taking those predictions of the tens of thousands of compounds we've looked at computationally, what subset should we actually buy or synthesize and test experimentally to see if it binds and inhibits this important target. So that's one of the things. And then we're hunting for these alternative structures of these proteins, as I mentioned, with the spike or demogorgon, as we like to call it, and starting to take those, essentially novel structures that no one has seen before, and gain insight into how these things work, and also use them as starting points for rational drug design. And we're starting to explore antibody design as well. So our strategy is try everything we can in parallel, and we'll see what sticks.
So that was going to be one of my other questions, which is like, you're producing all this information. I guess who's consuming what you're producing that would then be able to make a therapeutic against it, or are you trying to do both?
Great question. Yeah, so we are a publicly funded operation. Most of our funding comes from the NSF and NIH, and so we try to do things as in the open as we can. So in the past, we've always shared data sets upon request after publication. In this case, with the immediacy of the pandemic, we're making extra efforts to share our data as quickly and as broadly as possible. So we're engaged with a number of cloud service providers and are working on ways to put these data sets, which are pretty large, up on the internet for anyone who wants to dig into. And so our hope is that in much the same way as having more computers work on these problems in parallel accelerates progress, that making the data available and allowing more brains to crunch on it and see what value we can extract will also accelerate progress.
Yeah, AWS has now a data exchange platform that they've created for scientific data.
That's right.
Yeah.
That's right. Yeah. So we're talking with their public data sets team.
Okay. Yeah, I've been looking at one of my friends runs it, so he's been showing me sort of what they make available, which is interesting, of how they want to play that role of intermediary, or providing a system for everybody being able to exchange data.
That's right. Yep. So that's one of the projects, and there's a couple others that are also trying to help with this, which is great because we and others are producing enormous amounts of data, and it's not trivial to just put it up on our website, for example.
Yep. Well, and there's the production of the data, and then there's the analysis of it, which sort of, brings me to, like, so you said you're focused on the Demogorgon, right? The Stranger Things
that's right.
beast. But is that the protein that you're really focusing on? And have you started to understand its structure and mechanism of action, or where you think something can be targeted against it?
Yeah, so the spike complex and the main protease are some of our initial focal points. There are 28 proteins in the viral genome, and we have simulations up and running of about half of them at this point, as well as different complexes between them. So our hope is to, on the analysis side, focus our mental energy on a subset of them, starting with the spike and the main protease, and to get the data out there on the others for others to chew on as well. And we've been making a lot of good progress. So this past week, the first batch of experimental tests based on predictions coming out of Folding at Home were performed on the main protease, and there's some interesting leads in there that warrant further follow-up. So that's a really exciting step, just to have done the experiments, in addition to finding tantalizing hints that more progress is in the works. And we've been finding what we call cryptic pockets in the main protease. So these are really fascinating because often when you look at the experimental structures of one of these proteins, there are few or maybe no concavities or pockets in the surface where a small molecule drug is likely to be able to bind tightly enough to serve as a candidate for drug development. But often when we go and look at all the moving parts and what they're doing, we see these cryptic pockets that are absent in that starting
Right.
point open up. And we can often find that they actually will be quite valuable targets for drug discovery. So we're making progress on that front. And we've also already written some blog posts and tweets about actually seeing the opening of this spike complex and starting to understand what that looks like and what opportunities for things like antibody design that might open up.
Yeah, I mean, in one of my previous podcasts, I interviewed Jake Glanville about distributed bio, how they're, engineering the old SARS antibodies to
Right.
work against coronavirus. And I think I just saw that they were tweeting or talking about having some strong success where they've had 4 different laboratories test out the antibody against the spike protein itself.
Okay. Oh, that's great to
So
hear. I've enjoyed hearing some about their work. And
yeah, well, the sooner we come up with something to manage this disease, the better off I think everybody will be. But
agreed.
um, you know, when we talk about designing drugs against some of these proteins and why it's so difficult, can you sort of explain that just a little bit? I think it's difficult for people to understand that, if you've got the key, you need the hole where it can go in and unlock something, and sometimes that hole is not available. So
Mhm.
how would you describe that if you were walking somebody through why something may or may not work well?
Mhm. Yeah, this really gets at one of the challenges of working at this scale, which is that, at the macroscopic scales that we typically think about, life seems very deterministic. And maybe it's a little too strong to say predictable, but there's a lot of predictable aspects of our life, right? At these small scales, everything is very stochastic and very coupled, right? So if you see something happen once, it doesn't mean that's usually what happens. You could have just seen an outlier. And in fact, there's many different outliers. So you have to observe things many times to get a real sense of what typical behavior looks like. And further complicating things, everything is strongly coupled together, right? So if you're stuck at home on quarantine, you know that if you open your front door, that is independent of what's happening to your back door or your windows, for example. But in the realm of proteins, that's not true. Opening the front door might cause the back door to become impossible to open, essentially, or might cause it to spontaneously spring open and your windows to go up and down. And so it's really hard to predict what the effects of small changes like adding an atom to a drug-like molecule are going to be, because there's all kinds of responses that come into play that are often at odds with each other and make it very difficult to predict exactly what's going to happen. So there's a huge amount of trial and error in the drug discovery process. And one of the things that we ultimately want to do with Folding at Home is try to reduce that by having these quantitatively predictive models that let you make much more accurate guesses about how the system is going to respond to these small changes.
So essentially, the more and more people that jump onto your bandwagon, the faster and more effective you'll be able to look at these proteins and run simulations and come up with opportunities.
That's right. That's right. Yeah, we have a pretty insatiable thirst for more compute power, and scientifically, I would happily put all the world's computers to work on our problems, and I'm sure I would still run into things. I'm like, if only I had a little bit more, we could do such and such. But one of the really exciting things now is that the combination of technologies, including the computer hardware and the scientific code for simulating these things, have all, in recent years really come together that we can do a lot of really useful stuff that was completely impossible when I was a graduate student, and certainly before that. So, it's a really exciting time where computers have an unprecedented opportunity to accelerate biological and biomedical research as well as other fields.
Oh yeah, there isn't a day that goes by where I'm like, I need to go back to school. I mean, most of this stuff wasn't even like, I see high school students doing things that we weren't doing when we were in college, and I'm like, geez, wow, that's a totally different type of experiment. Um, so makes me feel like I always threaten. I tell my family, I'm like, I got to go back and get another degree. Like it's just uh, just so I can go and play with it myself. But so if you were to sort of walk somebody through the process of implementing this or doing this, how would you describe it to them? Since we don't have any screenshots or anything like that per se.
hmm so the process of like setting up and running one of these simulations, for
Yeah,
example.
I mean, if they were sort of to set it up on their end, and what do you see on your back end, I guess?
Right. So what we're doing on our side is taking these, experimental structures that give us just one snapshot of what this really complicated thing full of moving parts looks like, and we are surrounding it with water and other aspects of the solvent and environment that these things live in. And now we're taking this atomically detailed representation where, in very simplistic terms, you can think of there being a little sphere for every atom in the protein, and they're hooked up with springs to represent bonds, and they're pushing and pulling each other based on whether they're positively and negatively charged. So we hand this off to these molecular dynamics algorithms that are over and over and over and over and over again asking, given the ways these atoms are pushing and pulling on each other, where is each atom in this system going to be some small time in the future? And one of the reasons that this is so computationally expensive is that small time is a really small time, like 10 to the negative 15th seconds. And this is because, if we tried to work on longer time scales, we would get unphysical behavior like atoms jumping through each other, which just
Right.
doesn't make any sense. So in order to watch these tiny things collide and bounce off each other, we really need to work at these very, very short time scales. And then we need to do billions and thousands of billions and millions of billions of repetitions of this to build up to the timescales that are relevant to, biological systems and human life, where enzymes catalyze reactions and proteins change structures in response to light or odorants and all the other processes that we're interested in. So on our side, each of these, thousands of simulations that we might run of a protein on folding at home is kind of like a movie, right?
Right.
And as in a human interaction, you could start off in the same conditions, and every time you press play, something a little bit different might happen. And now what we can do is take all of this data and build a map of the space of possibilities. Again, returning to this analogy of, taking GPS data from lots of cars and using it to build up a map of this really large space that no individual car may have seen all of it.
Right. Right. So how do you see, the evolution of folding at home? I mean, where do you see it going in the next, year, 3, 5, etc.?
Oh man, I'm super excited about the folding future of folding at home. As long as we're still here, and so the project started almost 20 years ago now, and the initial focus was really on this very basic research question of how do many proteins start off as this extended linear chain of chemicals called amino acids and like fold up on themselves into these reasonably specific three-dimensional structures that can perform these amazing functions like catalyzing enzymatic reactions better than anything that we've come up with besides them, for example. And since then, the project has really evolved to broaden its scope. So initial steps, we're starting to think about misfolding diseases, protein misfolding diseases like Alzheimer's disease and Parkinson's and Huntington's disease. And then we started to grasp that these moving parts are really essential to the functions of many proteins. And we started thinking about, how do these things change in response to mutations implicated in cancer or Alzheimer's disease? And what can we learn from that about how these things work and how we could develop small molecules to fix them? And so looking ahead, I'm really excited that we do have this very general platform that essentially can be used to understand how anything that's small and made out of atoms works, which is quite a broad reach. So the initial focus will continue to be on different scales of organization in biological systems, but looking ahead, we also have the opportunity to think about, how can we draw on ideas from deep learning to analyze all this data, and how can we
Right.
apply these tools to material science. Things are super exciting.
Yeah, no, I mean, I've talked to a couple people in material sciences, and it's sort of interesting working on crystal structures and being able to sort of get an idea of what the next molecule should look like
Mhm.
before you've even made it. Um, there's some, computational and data is the capabilities that are coming online. It almost seems daily because I can't keep up with
That's
the
right.
publications that are coming out. Uh, Fundamentally going to change the way we do science.
No, definitely. I mean, there's a lot more opportunity for comparing and contrasting things and trying lots of things, right? So I mentioned that we've got simulations up and running of at least half of the proteins, I think, from SARS-CoV-2, but it's not just those like 12 or 14 proteins. We're also got simulations up and running of the variants of these proteins from the original SARS virus. So we can ask, well,
Right.
why is it that SARS-2 is so much of a bigger problem for humanity right now? And what insight does that give into how to deal with this? And comparing to other coronaviruses that have been around for a while, but not cause nearly as much of a headache, to understand what is it has changed. And this gets really exciting then also for considering large numbers of possibilities for drug and antibody design, where we can have some intuition guiding things, but also try lots in parallel, and after the fact asked which of these trials that we ran looks most fruitful.
Right. Right. Well, it's interesting, right? I mean, I think to myself that, not that long ago, it was sort of 80% lab and 20%, computer capability or IT. And now it feels like it's the reverse, where it's 80% computational capability and 20%, lab sort of work.
Yeah, I mean, and certainly the opportunity to combine them is one of the really exciting things. So my lab at this point is a third or half experimental, and we spend a lot of time hopping back and forth between these detailed insights that would be very hard to get by any other means besides our computer simulations, and then designing experiments to test these insights and to
Right.
test our ability to get in and manipulate things with small molecules or changes to the chemical composition of proteins, for example.
So, I mean, based on where you are, I mean, I'm sort of seeing a fundamental shift needed in the person doing the work,
Mm-hmm.
right? There's got to be an understanding on the comp side as well as the biological side coming
Mm-hmm.
together.
Mm-hmm.
Um, and I'm not sure we have graduated enough people in that combined domain yet.
Yeah, that is definitely a challenge. Is that within my lab, for example, we've got deep things going on with biology and chemistry and physics and machine learning and artificial intelligence and computer science and information theory. So I think one of the really exciting things is that we get to go and explore all these different regions of human knowledge, but it definitely also poses a challenge that it's a challenge to get sufficient grasp of all of these things. But this is one of the really interesting things with, is that makes it more and more important to work together in teams. And people need enough awareness of enough breadth to, help appreciate the opportunities, but then to go deep in their different subdomains and be ready and willing to work as a team.
Yeah, no, I think that, where we see the next breakthrough is this sort of cross-functional teamwork coming together.
Mm-hmm.
But I also think that labs like yours are great places to go and find people to hire.
That's right. That's right.
So, well, I mean, I can only wish you incredible success. The sooner we come up with a way to tackle COVID, I guess maybe we should ask people to jump onto folding at home and, put their computer available so that you can get that one more online to help you do more and more work.
That's right. That's right. Every little bit helps. And, one of the messages I'm always trying to get out there is that folding at home is about more than, just COVID. Certainly, this is the most important problem immediately. But looking ahead, we do have this very general platform. And I hope people that jump on board with helping with our COVID-19 work will recognize the value of applying the same approach to Alzheimer's disease and Ebola virus and all of these other problems, and will stick around with us and help us prioritize those in the future.
That's a good question now that you brought that up. How do people know that their contribution is making a difference?
Yeah, we do a lot to engage with our community. So we publish scientific papers, and we also write blog posts that try to be more accessible to the general public. And we're very active on social media, so Twitter and Facebook, for example. And we have a number of active communities where people can get engaged. So we have a forum at foldingforum.org that's mostly focused on helping people with technical issues related to participating in folding at home. And we recently started a Discord server, which people can go with their technical questions too, but that's also more of a forum where people can discuss with each other and with members of our scientific team to try to understand what's going on and what opportunities are out there.
No, that's a great way to extend someone's already existing education or just knowledge.
That's right. That's right. It's a really fun STEM opportunity. People are very hungry, especially now, to understand what the principles behind our work are and what we're doing, and what the results are looking like and the potential implications. And it's uh, uh, really really exciting to see those levels of engagement and interest.
Well, excellent. I wish you incredible success with what you're doing. And like I said, the faster and sooner that we get there, the better.
That's right. Thanks so much.
Thank you.
Thanks to all our volunteers. This has been tremendously helpful.